Today, this lecture for English speaker students. Arabic speaker students will be next week. So if you like to stay with us, so you can stay, but your class will start from next week. We talk about the cell injury, our first chapter in general pathology, cell injury. And we said last time that we have 4 features of cell injury. Reversible cell injury, irreversible cell injury, deposits, and adaptation. And last time I preferred to start with adaptation, and we explained adaptation, what does it mean, and how and when the cell can adapt with cell injury or any stimulus. Today, we will talk about the reversible and irreversible cell injury. And we will take today we'll discuss what does it mean and what the difference between reversible cell injury and irreversible cell injury. So our class today about the cell injury, reversible and irreversible. So cell injury. But before we talk about the cell injury, we have to talk first about the cell. What are the normal components of the cell and what are the normal function of the cell? How the cell can function normally. And after that, we will talk about the mechanism and the causes of injury and when we can say that this injury is reversible and when we can say that this injury is irreversible. So first, I would like to start with the normal conditions of the cell, the normal component of the cell, and how the cell can function normally. This is a cell. Nucleus. And here we have some organelles. So we know that this is cell membrane. And here the nucleus. And these are organelles. And of course, here we have DNA. And this is cytoplasm. All organelles are embedded in cytoplasm, which is the fluid of the cell. All these organelles and nucleus need energy. And we know that energy is ATP. The currency of the energy inside the cell is ATP molecules. So these organelles need ATP molecules to start to do their normal function. How the cell can get that ATP and how the cell can get its nourishment and nutrients, a lot of things. The most important thing, one is oxygen. So the cell need oxygen to produce ATP, to use ATP inside the cell. So the cell need oxygen to do its normal function, to produce energy. So what the problem, why this cell can be injured? Why the cell can get injury? A lot of causes of cell injury. The most important one, that the cell cannot get oxygen. The cell cannot get oxygen. So the cell will not be able to produce ATP. The cell will not be able to produce any energy and use energy to do the normal function. So the first cause is hypoxia. Hypo means decrease, oxia means oxygen. So the oxygen which go into the cell is decreased. Second, maybe infection. Infection can cause injury for the cell. Maybe microorganisms like bacteria, virus, fungi, parasite can injure the cell membrane or can injure any organ in the cell. Maybe your immune system. We will talk about a very big category of the immune disease called autoimmune diseases. Auto means self. Immune, it means that the immune system attack your own cells. So maybe the immune system start to produce immunoglobulin, which can attack the cell membrane, the nucleus, the organism, and can cause injury for the cell. Maybe some types of immune diseases called hypersensitivity. Hypersensitivity means when some microbe or bacteria, virus, fungi enter your bloodstream, so your immune system will react to this foreign body, but the reaction of the immune system will be exaggerated. It will be so much. So it's called hypersensitivity. Hypersensitivity diseases. So immune system can injure your cells. Maybe injury of the cell can be caused by physical causes like temperature. When you touch something hot, that temperature, high temperature, can cause injury for your skin cells. 5, maybe genetic. What does it mean, genetic? We will talk a very big category of disease called glycogen storage diseases. Glycogen storage disease mean that a lot of glycogen will be accumulated in the cells, and the cell will not be able to produce these glycogen out of the cell. So glycogen will be accumulated inside the cell and will cause injury of the organisms inside the cell. Maybe also nutritional. Nutritional means that your cell needs to eat proteins, lipids, carbohydrates to do their own function. So your cells need to eat. Some nutritional disorders, like we talk about some of them last week. It's called marasmus. Marasmus means severe macromolecules deficiency, like severe protein deficiency, lipid deficiency, and carbohydrate deficiency. So there's no protein, there's no lipid, there is no enough carbohydrates to the cell. So there's no appropriate nutrients for the cell, so the cell can get nutritional cell injury. But the most important one, and we will talk about it today, is hypoxia. Hypoxia is the most important one. So now we will talk about the mechanism of hypoxia, how hypoxia can cause injury for the cell, and if the cell get injured, how this injury can be reversible or irreversible. So today we're gonna talk about the most important stimulus of cell injury. It's called hypoxia. So we will talk about the causes of hypoxia, causes of hypoxia, how hypoxia can cause cell injury and how it can be reversible or how it can be irreversible. What the difference between them? First of all, causes of hypoxia. Hypoxia means there's not enough oxygen is going to the cells. There's not enough oxygen going to the cell. Decrease oxygen supplement to cells. Why oxygen is decreased to cells? How oxygen can go to the cells in the first place? Oxygen is carried on red blood cells. Inside red blood cells, we have very important protein. It's called hemoglobin. Hemoglobin carries oxygen inside the red blood cells to the cells. So if we have decreased in hemoglobin, so we will get hypoxia because oxygen is carried on hemoglobin. So what does it mean, decrease in hemoglobin or decrease in red blood cells? It means anemia. So if you have anemia, so it means that you have decrease in red blood cells, decrease in hemoglobin amount. So the number of oxygen is carried to the cell will be decreased. So anemia can cause hypoxia. Also, another cause of hypoxia and more dangerous, it's called ischemia. What is the difference between ischemia and anemia? The difference that anemia, it means that the blood is going to the cells, but not carried with enough oxygen. The blood is going, but there is no enough hemoglobin or there's no enough red blood cells is going to the cells. But the blood is going, but without enough oxygen. But ischemia, it means there is no blood is going to the cell and there's no oxygen. So for example, this is the cell and here is the blood vessel. Anemia, it means that red blood cells is going, but without enough hemoglobin. So without enough oxygen. But ischemia means that there's something is blocking the blood vessel. So there is no blood is going. So there's no oxygen is going to the tissue. And both of them, of course, cause hypoxia because either anemia or ischemia, there is no enough oxygen is going to the cell. So the main causes of hypoxia, anemia, and ischemia. Okay, but how hypoxia can cause cell injury? Why the cell can get affected if it doesn't get enough oxygen? What is the importance of oxygen inside the cell? That's the question. So, mechanism of hypoxia and cell injury. How hypoxia and why hypoxia can cause cell injury. We know in biochemistry, we talk in biochemistry that to produce a lot of ATP, you need oxygen. You took some process called aerobic glycolysis and another process called anaerobic glycolysis. Aerobic means air, it means there's enough oxygen. Anaerobic, it means there is no, and means no, there is no oxygen in cells. And we know in the chemistry, in aerobic glycolysis, in presence of oxygen, each molecule of glucose will produce more than 38 ATP. But In anaerobic glycolysis, the cell can produce only 2 ATP. So the main effect of hypoxia on the cell, that hypoxia leads to decrease of ATP. Okay, but what's the problem? What's gonna happen if there is no enough ATP inside the cell? Also, you studied in physiology, very important topic. This is called sodium potassium ATPase pump. What does it mean, sodium potassium ATPase pump? In the normal condition of the cell, sodium should be outside the cell and potassium should be inside the cell. Calcium should be outside the cell, magnesium should be inside the cell. These ions, potassium and magnesium, normally they are found inside, intracellular, inside the cell. Sodium and calcium should be outside the cell. So the main important of this pump, when inside the cell there's much sodium, this pump will try to get out sodium and taken potassium inside. So the main importance, the main rule of sodium potassium ATPase pump to take sodium outside the cell and put potassium inside the cell. But why we call sodium potassium ATPase pump? It means this pump to work, probably to work normally, needs ATP. It needs ATP. So if you don't have enough ATP, so this pump will not work normally. So the sodium will increase inside the cell. Okay, but what's gonna happen if sodium increase inside the cell? Two problems. Two problems if sodium is increased inside the cell. First, we know that sodium has osmotic pressure. What does it mean, osmotic pressure? It means that sodium can draw the water inside the cell. So if sodium is increased inside the cell, so sodium will draw a lot of water inside the cell. So sodium is increased. Sodium is increased. So sodium will draw a lot of water inside the cell. So the water will start to get swelling. The water will start to get swelling. So very important organelle inside the cell, it's called mitochondria. Mitochondria start to be swelling. A lot of water will get inside the mitochondria. And we know that the mitochondria is the factory of energy inside the cell. So mitochondria will start to get swelling. A lot of water will start to get inside the mitochondria, and mitochondria will be destructive. So mitochondria will damaged. What's the problem of mitochondria damaged? First, there is no enough ATP, so the pump will not work, and a lot of sodium will stuck inside the cell. And second, mitochondria, we know that inside the mitochondria we have a lot of free radicals molecules. What does it mean, free radical molecules? Free radical mean that some molecules, they have active oxygen atom. But what's the problem with active oxygen? The active oxygen can bind to protein inside the cell, can bind to carbohydrate and lipids inside the cell and start to make oxidation of these molecules and destroy all proteins inside the cell and all lipids. And we know that the cell composed of protein, lipids, and carbohydrates. So free radicals, when be released from the mitochondria, will destroy all the cell. And also, we inside the cell we have some inside the mitochondria we have some important enzyme. It's called cytochrome C. Cytochrome C is a very important enzyme, but inside the mitochondria. If cytochrome C released out the mitochondria, so this enzyme will start to activate a very dangerous process called apoptosis. What does it mean, apoptosis? Apoptosis means programmed cell death, suicide of the cell. The cell will kill itself. And we will discuss this topic in detail lecture. So again, there is no oxygen, so there is no ATP. There is no ATP, so sodium, potassium pump will not work. What's the problem? This is the cell. If sodium, potassium pump will not work, what was the work of sodium potassium pump? The work was to take out sodium and put in potassium. So now sodium will not get out. Sodium will be accumulated inside the cell. Sodium will draw inside a lot of water inside the cell. The water will start to get swelling. The water will get inside the mitochondria. The mitochondria will start to be damaged. The problem of damage of mitochondria, we have three problems. First of all, mitochondria is the main source of ATP. So there is no ATP. So we will get more and more decrease in ATP. Second problem, that inside the mitochondria we have free radicals. Free radicals, they have active oxygen atom. What's the problem with active oxygen atom? Active oxygen atom can bind to protein, lipids, and carbohydrates inside the cell and start to make oxidation of these molecules and start to destroy these molecules. And also we have very important enzyme, it's called cytochrome C inside the mitochondria. It's a very important enzyme. It has a very important role in a lot of metabolic process, but inside the mitochondria. So if it get out of mitochondria, so it will start to activate very dangerous process called apoptosis, programmed cell death. It will stimulate the cell to kill itself. That's the first problem. So is it clear? So we don't have enough oxygen, so we don't have enough ATP. So there's not enough ATP. Sodium potassium pump will not work normally. So sodium will accumulate inside the cell. Sodium will draw in a lot of water. Water will cause cell swelling. The water will get inside the mitochondria. Mitochondria will be damaged. After damage of mitochondria, we have three problems. There is no enough ATP because of course the mitochondria is damaged. And second problem, free radicals inside the mitochondria will be released in cytoplasm and start to destroy all organs and cell membrane. The third problem, that cytochrome C, it was a very important enzyme inside the mitochondria. If it releases from mitochondria inside the cytoplasm, it will start to activate a very dangerous process. It's called apoptosis, programmed cell death. So until now, everything is clear? Anyone has a question? Okay, so we can move to the next step. Next problem, If ATP is decreased, also we have a problem with calcium. What we do have with calcium? Do you remember when I told you that sodium potassium pump will not work? So sodium will increase inside the cell and will get a lot of water, yeah, and also will stimulate a lot of calcium to get inside the cell, because sodium open some channels. It's called sodium calcium channel, and get a lot of calcium inside the cell, because according to the normal concentration of calcium outside and the inside, we have 10,000 times calcium outside more than inside. So when we open the channel, a lot of calcium get inside the cell. Okay, but what's the problem with calcium? What's gonna happen if calcium is accumulated inside the cell? What's the problem? The problem that calcium does very dangerous process. Calcium can activate very dangerous group of enzymes. It's called damaging enzyme. Damaging enzyme. Enzyme which can damage all molecules and all organs inside the cell. Like what? Example. First one, we have some enzyme intercellular. It's called ATPase. And of course, from its name, you can understand the function. ATPase means enzyme which can break down ATP. So we have more decrease in ATP. Second, we have enzyme called proteases. Calcium can activate these enzymes, and from its name you can understand the function. Proteases, so enzymes which can break down proteins inside the cell. And of course we know that protein is a very important molecule, very important composition of cell composition. And also we have an enzyme called endonucleases. These enzymes can nucleases, it means can break down the nucleus of the cell. Number 4, we have enzyme called caspase. Caspase enzyme. And caspase enzyme can activate apoptosis like cytochrome C. So caspase can activate programmed cell death, apoptosis procedures. 5, also calcium can activate a group of enzymes, very dangerous, and the most dangerous enzymes between them, it's called phospholipases. Phospholipases. And we know that the cell membrane, this cell membrane, we studied in histology that the cell membrane is consisting of phospholipid bilayer. Bi means two. So the cell membrane of the cell and all organelles and the cell membrane is consisting of phospholipid bilayer. So it means that when calcium is increased inside the cell, calcium will activate a lot of phospholipase enzymes, and phospholipase enzymes will destroy all cell membrane of the all organelles inside the cell, and will destroy the cell membrane of the cell itself. So that's why the most dangerous enzyme between all of them. So again, it's all about decrease of oxygen. Decrease oxygen lead to decrease in ATP. Decrease in ATP means sodium potassium pump will not work anymore. So it means that the The main rule of sodium potassium pump to get sodium outside the cell. So now sodium will not go outside. So sodium will be increased inside the cell, and sodium has two main rules. Sodium will increase the water inside the cell and cause cell swelling, and also sodium can increase calcium inside the cell. And each of them is a very, very dangerous problem. Why? Cell swelling can lead to mitochondria swelling. So it means that mitochondria will be full with water and mitochondria will be ruptured and damaged. So if mitochondria is damaged, of course, there is no ATP. So we will get more and more decrease in ATP. Also, inside mitochondria, there are free radicals. They are safe, but inside mitochondria. So if they get out of mitochondria, so they will start to make oxidation of all composition of the cell and will destroy all the cell. And also we have cytochrome C enzyme. It's a safe and very important enzyme, but inside the mitochondria. So if the mitochondria is damaged, cytochrome C will be released in cytoplasm and will start to activate a very dangerous process called cell apoptosis. And calcium, the main dangerous point of increase intracellular calcium is that calcium can activate damaging enzymes. Damaging enzymes like one, ATPase. ATPase destroy a lot of ATP. So we get more and more depletion and decrease in ATP. Second, protease. Calcium will activate protease enzyme, so decrease and destruction of proteins of the cell. And we know that protein is a very important composition of organelles and the cell. Three, caspase enzyme. Caspase will activate apoptosis. Apoptosis means programmed cell death. And don't worry, we will explain apoptosis in a separate lecture. Oh, endonucleases. And endonucleases will destroy the nucleus. And of course, the cell cannot live without nucleus. 5, phospholipases. And this is the most dangerous enzyme between them. Why? Because phospholipases can destroy phospholipid. And we know, of course, that cell membrane of the cell and all organs and the nucleus is composed of phospholipid bilayer. So these enzymes will destroy all organs and all cells. That's why it's the most dangerous one. Let me talk a little bit about endonucleases and phospholipases. First, endonucleases. It means the enzymes which destruct the nucleus of the cell. This is the nucleus. The nucleus of the cell, and here we have DNA. When endonucleases start to destruct the nucleus of the cell, first of all, these enzymes will start to destruct the membranes of the nucleus, and cause, see, the nucleus start to decrease in size. Decrease in size. It's called pyknosis. Pyknosis. It means that the cell will start to condensate. Condensation. Condensation means that the enzyme will start to decrease the size of the nucleus. After that, after that, the enzymes will start to destroy the nucleus into some fragment. So separated fragments of the nucleus. It's called karyolexis. Karyo means nucleus. Lexis means breakdown or fragmentation. So here, the nucleus is destroyed and fragment, a lot of fragments. After that, the enzyme will start to destroy all these fragments, or and the nucleus will be disappear. So it's called karyolysis. So 3 steps: pyknosis. It means condensation. Condensation means decrease in size of the nucleus. After that, karyolexis. It means fragmentation. It means it will destruct the nucleus into 5 or 6 fragments. 6 parts. Fragments means parts. After that, karyolysis. It means disappear of nucleus. So endonucleases can cause pyknosis, karyolexis, and karyolysis. Second one is phospholipases enzymes. There's the cell, and if we take some part of the cell membrane, we will find that the cell membrane is composed of phospholipid bilayer. There's one layer and there's another layer. So 2 layers means bi. So it's a phospholipid bilayer. What's the problem with phospholipase enzyme? When phospholipases come and start to destroy the phospholipids, we destroy the phospholipid into fragments or parts. These parts will get inside the cell. So this is called myelin figures. Myelin figure is the remnant of destroyed cell membrane. What's the problem with these myelin figures? These figures in normal cells, these figures should be phagocytosed. It means macrophage will come and engulf these figures. But in abnormal cells or in diseased cells, these myelin figures will bind with calcium, And when these remnants of destroyed cell membrane binds with calcium, so they will collect together and form very heavy substances called soap and cause something called some procedure called saponification. Saponification means forming of soap heavy substances, and this substance the cells cannot get rid of this substance, and this substance will accumulate inside the organs, inside the nucleus, and start to kill the cell. So again, the cell injury, the most important cause is hypoxia. But why we can get hypoxia? Because of anemia or ischemia. Okay, but what's the problem with hypoxia? Hypoxia may lead to decrease ATP. The problem with ATP that when ATP is decreased, we have very important pump inside the cell called sodium potassium ATPase pump. This pump will not work, so sodium will be increased inside the cell, which is abnormal, and sodium can lead to two problems. First, sodium will draw inside a lot of water, and also sodium can open some channels called sodium calcium channel, and we know in physiology that calcium extracellular outside the cell is 10,000 more than inside the cell. So when we open the channel, of course, a lot of calcium will get influx inside the cell. So sodium will be increased inside the cell. There's a problem. This is a disaster, very dangerous problem. Water can cause cell swelling. Cell swelling to mitochondria swelling. The water can get inside the mitochondria, and the mitochondria will damage. So when mitochondria damaged, so there is no enough ATP, so we will get more and more ATP depletion and decrease in ATP. Second, inside the mitochondria we have some dangerous molecules called free radicals. Free radicals contain active oxygen. Active oxygen can cause oxidation and breakdown of protein, carbohydrates, and lipids, all composition of the cell. And also inside the cell, we have very important enzyme called cytochrome oxidase C. This is important, is a very important enzyme inside the mitochondria, but when it released out the mitochondria, it start to activate dangerous process called apoptosis. And of course, in calcium, the very dangerous role of calcium, that calcium can activate damaging enzymes. Damaging enzyme like ATPase, proteases, caspases, phospholipases, endonucleases. So we talk about two mechanisms or two causes of decreased ATP. So when ATP is decreased, can cause problem with sodium, potassium pump, and sodium can increase the water inside the cell. And also, when decrease in sodium and ATP, sodium potassium pump will not work, and that will lead to increase in calcium. Last mechanism of decrease ATP, when the cell doesn't have enough ATP, so the cell will start to go to something called anaerobic glycolysis. Glycolysis means lysis means breakdown of glucose, and we know the cell, to produce energy, the cell will break down glucose. But normally the cell break down glucose in presence of oxygen. It's called aerobic oxygen. But now the problem that we don't have oxygen, so the cell will start to break down glucose, but in absence of oxygen, and it's called anaerobic glycolysis. But what's the problem with anaerobic glycolysis? The problem that glucose will breaking down into very bad substance called lactate or lactic acid. Lactic acid. What's the problem with lactic acid? It's acid. So it can cause acidosis of intracellular environment. But what's the problem with acidosis inside intracellular environment? The problem that acidosis will increase or activate damaging enzymes. What the heck? So this acidosis also will do the same like the calcium do with damaging enzymes. So now we talk about mechanism of cell injury with hypoxia. Okay, but how we can differentiate between reversible and irreversible? How we can tell that the cell can come back to its nature and normal structure, or can't? It's called point of return. Points of return. It means what factors or how the cell can come back, return back to the normal structure. If the destruction was in cell membrane destruction, or mitochondrial destruction, of course it's irreversible. So if these causes lead to cell membrane destruction or mitochondrial destruction, so the cell cannot live anymore. So here the steps: decrease oxygen leads to decrease ATP, leads to sodium potassium pump will not work, so sodium will increase, and that will increase water and calcium. So we have to stop the injury in some step in a specific step. Like for example, we can solve the problem here. We can increase the sodium supplement, or maybe we can increase the ATP concentration, or maybe we can increase the work of sodium potassium pump. But if the injury reach to the cell membrane or mitochondria membrane and lead to destruction of cell membrane or mitochondria membrane, so the cell will not come back to its normal structure anymore. So the point of return, how we can distinguish between reversible and irreversible, it depends on the location of destruction. If the destruction happened in cell membrane or in mitochondrial membrane, so unfortunately the cell will die because the cell cannot live without cell membrane, phospholipid cell membrane, and mitochondria. Because we know when mitochondria damaged, there's no ATP, and the cytochrome oxidase enzyme will release and activate apoptosis, and all free radicals inside the mitochondria will be released and destroy all the cell. So if the mitochondria destroyed, so the cell will not live anymore. If the cell membrane is destroyed, so the cell cannot come back to its normal structure. So it's irreversible. Otherwise, it's reversible. Is it understood? Anyone has a question? So I will open the mic. If you have any question, go ahead. Guys, do you have any question? Yes or no?
No.
Okay, so everything is clear? Okay, so we finished for today.
Yes.
And yeah, go ahead. Who's asking? Okay, guys. So let me first stop the record.